All right, so we're going to move on to chapter 7 then, which is membrane structure and function. So we've talked a little bit about membranes already, haven't we? Yep, the plasma membrane in particular, and all of the other parts inside the cell that are bounded by membrane or when they're made of membrane. So can you think of some things that are inside the cell that are where the membrane either surrounds them or the membrane itself is the structure? Not the cell membrane, but where membrane. Nucleus, nuclear envelope, yep.
Do you have proteins in the membrane?
There are proteins within the membrane, yep. Golgi apparatus, yep. Mitochondria, chloroplast, the ER. Ribosomes are not. Remember, ribosomes are big molecules made of RNA and proteins. Okay? So there's a lot inside the cell which is made of membrane. We're going to focus on the plasma membrane that surrounds the cell. All right? Okay, so if you were to go look at a real, like the original of this, where would you have to go? Egypt, yep. You'd have to go down into bottom of one of the pyramids, or actually, no, not, well, I don't know if these are actually in the pyramids. I didn't go inside the pyramids. I went to the Valley of the Kings and Queens, where you could go down and see where they were buried. And these kind of paintings are lining the tunnels that go down underneath.
Oh, so that's an actual photo you took in there.
So I can't remember if this is one of my photos. I don't think it is. I think I ripped this one off. I think when I went there, it was a long time ago, and it was all film pictures. So, and even though I scanned them in, it was a hassle to look through them. It's quicker just to grab it off the internet. And also, they don't let you use flash photography. They're really fussy. You can't use a tripod, and it's dark down there, right? So you've got guards. You've got tomb guards all over the place. Yep. And it's a little bit sketchy, at least when I went it was. And so you could take handheld photographs of the walls, but it's really dark. So you're worried about them coming out blurry. That's why people want a tripod. But if you've got a tripod, then they charge you, right? I didn't want to pay the extra money to do it. So I thought I would get away with it by doing this, right? You ready for this? This is like a grey area. You know what a monopod is? So I thought, if I have a monopod and I put it on my foot, it's not on the ground and it's not a tripod. Their rules are a tripod. So I was down in the monopod taking pictures, and one of the tomb guards come up and he just went like that. And I said, come on, it's not a tripod, it's on my foot, it's not even on the ground. And he went like that, and I continued taking some pictures. And then he pulled back his robe, and he had a big, dirty, great big knife underneath it. And I said, all right, I won't take any more pictures.
When did you go?
This was in year 2000.
Because now, I went 2 years ago.
Yeah.
It didn't let us take pictures.
None at all?
We had to leave our cameras behind.
Wow. All right, so it's changed quite a lot then. All right. Not even if you pay for a permit?
You probably could do that, but we didn't want to do that.
Yeah. All right. So anyway, why have I taken you back to ancient Egypt?
Ancient Egyptians were masters of medicine.
They were. They were obsessed. Not obsessed, but a very important part of their life was getting to the afterlife, right? Once they died, here's the land of the living, they wondered where the land of the dead was. Where do they go? What's the journey to the afterlife? Pyramids were built so that the pharaohs could have enough for their journey to the afterlife, right? So that begs the question, I guess, what's the distance between the living and the dead? The ancient Egyptians didn't really know, but they felt it was this big, long journey. What do you think is the distance?
Between the living and the non-living?
Yeah. What is that distance?
Length of a pulley.
Sorry?
Length of a pulley.
Length of a pulley?
A bullet.
A bullet? Ooh, all right. 6 feet under? I don't know. By how far are they separated? It's a bit of a philosophical question, maybe, isn't it?
Because everybody dies and lives at different times. Not everybody dies at the same time.
They don't.
So
Well, if we're talking more on biological level, they're right next to you.
I'm going to give you a very literal answer to this question, right? I'm going to give you a distance. It's about 8 nanometers. That's the distance between the living and the non-living.
It doesn't make sense. I have to
Yeah, how do you measure that?
How can you measure it?
distance.
Well, think about it.
It's your cells right next to
Here's a cell.
Oh, the rate of when they live.
That's the basic unit of life, the cell. So if I'm outside the cell, then I'm outside of that basic unit of life. And the thickness of the membrane that separates the inside to the outside is about 8 nanometers.
Oh, so you're talking of biological though.
Okay. You okay with that? All right. I don't think the ancient Egyptians would buy it, but anyway. All right. So let's have a look at the plasma membrane then. All right. Firstly, we're going to have a look at the structure of the plasma membrane. So think about the plasma membrane. That's the boundary, all right, that separates the living cell from its non-living surroundings. Okay? All of life processes pretty much conducted inside the cell. Outside the cell, you might still be in the living organism, but you are outside of the cell. And it's the plasma membrane which is the boundary that separates those two. And it's about 8 nanometers thick. Now, to give you an idea how thin that is, it's about 10,000 times thinner than a piece of paper. Does anybody remember what nano is? Billionth. It's a billionth of a meter. Nanometer is 10 to the negative 9. All right, so it's 8 times 10 to the negative 9 meters. So it's 8 1/billionths of a meter. Okay, that's what a nanometer is. Is everybody okay with that? We're going to talk about nanometers now because we're sort of looking at that scale of big molecules and membranes. All right? And the plasma membrane, it exhibits something called selective permeability. Thank goodness. That means some substances cross it more easily than others. Some substances can cross easily. Some substances cross with a great deal of difficulty. And there are lots of mechanisms as to how the cell regulates the movement of substances in and out of the cell. But everything that goes in and everything that comes out has to go through the plasma membrane. All right? It's a bit like border patrol for the cell. Okay? That's what it is. So, phospholipids, and we've talked about phospholipids, haven't we? Do you want me to give you a quick refresher on what they are, how they're structured? Or do you remember?
It's the
hydrophobic tail.
That's it?
hydrophilic head.
We've got a hydrophilic head, which is a big phosphate group. That's connected to a glycerol molecule. And then you've got 2 fatty acid chains attached. Now, why have I drawn a little kink or a bend in that fatty acid chain?
One's unsaturated.
Right, that would be the location of a double bond. This one's unsaturated, this one's saturated. About how many carbons long are these fatty acids? About?
16.
Yeah, 16, 18-ish, plus minus. Okay, so is everybody okay with that? And I'm going to draw it and you'll see it represented kind of like this. A bit like that. Okay? The phosphate head is hydrophilic,
Philic.
which means water loving. And the fatty acid tails are?
Hydrophobic.
And why are they hydrophobic?
It's because it's a lipid. It's a fat. It's
The carbon.
Primarily non-polar covalent bonds, right? Good, good. Okay, so the most abundant molecule in the plasma membrane are phospholipids. All right, that's a major component of the plasma membrane. And phospholipids are amphipathic. Now, we've just described what amphipathic is. They've got a hydrophobic region, the fatty acid tails, and a hydrophilic region, the charged phosphate head. Okay? Now, the membrane itself, you can think about it as a fluid mosaic. That's what it's referred to, as a fluid mosaic. So it's kind of like a lake of phospholipid, thin lake, with proteins suspended in there. And they kind of bob around and move around. Okay? Is everybody okay with that? At a very sort of basic level.
It's referred to as what?
Fluid mosaic. Yep. You've got this like thin lake of phospholipid, and it forms a phospholipid bilayer. I don't know if you remember that. And then there are proteins embedded in that phospholipid bilayer, and they kind of bob around and float around. They're mobile. So it's fluid, but it's a mosaic. All right. So if we were to look at a cross-section of the phospholipid part of the membrane, then here you've got your hydrophilic head, there are your tails, and here you've got another layer of phospholipid, all right? And it forms this phospholipid bilayer, 2 layers, okay? And out here you've got an aqueous environment. This might be the outside of the cell, and here you've got an aqueous environment which is the inside of the cell, okay? And I think we talked about, when we talked about phospholipids, why they arrange themselves in this way, yeah? Good.
So
Alright.
the proteins, like,
So I'll show you the proteins in a moment. So proteins,
they were on the video, right?
they were on the video, yeah? Proteins were on the video, yeah? So I'll show you how the proteins sort of configure themselves into the phospholipid bilayer. All right, is everybody okay with that then? This phospholipid bilayer, yeah? Okay, good. All right, so you got the phospholipid bilayer, and there are proteins embedded in that phospholipid bilayer that kind of bob around and float around. They're mobile, they're able to move, they're not static. Most of them. Some of them are locked into place, but most of them move around. So that's what the fluid mosaic model of membrane structure states or describes it as. All right. Now, the proteins aren't randomly distributed in the membrane. Okay, so they can be concentrated into areas, they can be next to one another. So it's not random. There's a very specific, I guess, reason or design, not design, but there's very specific ways in which they're organized within the membrane. Yeah. It's not random. One might be next to another, which might be next to another, which might be next to another, and they're organized in that way because maybe it's a chain of enzyme molecules, or they're arranged because they hold cells together. But there's a very distinct pattern to how they're arranged. It's not random, even though they can move, many of them. All right, so let's have a look at then this diagram, and it shows you, it's a really nice picture of the membrane. So in here, we've got the inside of the cell, cytoplasmic side of the membrane, and here you can see microfilaments of the cytoskeleton. Out here, we've got the extracellular side of the membrane, where we've got the extracellular matrix, and we've got water also. So it's an aqueous environment outside with the extracellular fluid, and an aqueous environment inside with the cytosol. These are our phospholipids forming the bilayer, and here are the proteins that are wedged or sort of embedded within that phospholipid bilayer. All right. Now I'm going to describe these proteins in a bit more depth. For example, you've got a protein like this, which is only on the outside, doesn't really penetrate into this hydrophobic part of the phospholipid bilayer, and that we would call a peripheral protein. And then we've got some proteins that, like this one, that penetrate into the hydrophobic part of the phospholipid bilayer. And then we've got proteins like this, which have a little bit on the outside. They penetrate through the membrane, and they would be called transmembrane proteins. So we've got peripheral proteins, we've got integral proteins, and then we've got ones that span the membrane, which are called transmembrane proteins. I'll describe them in more depth later. I'm just trying to give you sort of an idea of the landscape we're dealing with. So can you see all these proteins, the way they're sort of arranged? There's a group there, there's a group there. What could this be?
Carbohydrate.
That's a carbohydrate chain attached to a protein. So what would that whole thing be? A carbohydrate chain attached to a protein. Was it? good, glycoprotein. Now sometimes the carbohydrate chains are attached to the lipids, in which case we've got a glycolipid. All right. Now the other part I want you to pay attention to is right here, cholesterol molecules. Cholesterol molecules are steroids, and they're wedged in here in the phospholipid, in the hydrophobic part of the phospholipid bilayer. Okay, so we've got our phospholipid bilayer, cholesterol molecules wedged in there, but in the hydrophobic portion. We've got proteins embedded, some of them only on the outside, some that penetrate in, and some that span the whole membrane. And then we've got carbohydrates, some of which are attached to proteins, and some of which are attached to the lipids. Is everyone okay with that? Those major different components. They're the primary things I'm going to talk about, and not much else. Okay? Good? And all this can move quickly, dynamically. All right. So, let's define peripheral proteins then. Peripheral proteins are bound to the surface of the membrane. They don't penetrate into the hydrophobic part of the phospholipid bilayer. So they're bound to the surface. Integral proteins is any protein which penetrates into the hydrophobic part of the phospholipid bilayer. So if any part of the protein is into that the hydrophobic part of the phospholipid bilayer, it would be called an integral protein or an integral membrane protein. And then proteins, integral proteins that span the entire membrane. They've got a little bit that sticks out on the extracellular side and a little bit that sticks out on the inside of the cell. We call those transmembrane proteins. All right, so what we're going to do, I want you to make a little sketch of the membrane. All right, and again, these little sketches are really powerful learning tools. They usually take you just 1 or 2 minutes or sometimes even 30 seconds to do. You can label them and you can do it anytime waiting at traffic lights. I don't know, when you're just sitting in front of not doing very much. When you're out the shower. When you're in the shower, just, let the glass door steam up and draw it with your finger, right? Loads of times when you can do these little sketches. All right, so let's draw the phospholipid bilayer. Again, I'm trying to be quick. All right, what would we label these? Each one of these.
Phospholipid.
Good, phospholipid. And what part of the phospholipid is this?
Hydrophilic.
It's the phosphate head. Its property is hydrophilic, yeah? And this would be the
tails.
fatty acid tails, and their property is
Hydrophobic.
hydrophobic, good. All right, so now I'm going to put a great big what? What would this be? A protein. What kind of protein is it? You've got a choice, peripheral or integral? Integral. What kind of integral protein is it? Transmembrane. Good. I'm going to draw another little protein just there. And I'm going to put another one just there. Okay, so let's make sure we can label all of these. What would you label this protein? Peripheral membrane protein. What would you label this one? Integral proteins. Transmembrane? No. And what would you label this double layer of phospholipid? Phospholipid bilayer, good. Now just looking at this right now, could you tell which side is the extracellular side and which side is the intracellular side? No, you can't. Okay, good, good. Okay, so here's a nicer diagram than I can draw. It just shows one of these transmembrane proteins, and you're familiar with protein structure, aren't you? Yeah, it just shows how, just gives you some detail of the protein structure. Okay, and that's kind of important as to which parts of the protein are on the outside, the more hydrophilic parts, and which parts are the more hydrophobic parts of the protein. Okay? All right. Okay. So everybody's okay with labeling everything on here. Is there anything I left out? Oh, is there a couple of things I said I'm going to talk about these, but I totally left them out of that diagram.
The chloroplasts.
Which?
Distinguishes chloroplasts. Or cholesterol, sorry.
Good. Cholesterol. Where am I going to put the cholesterol?
Inside.
I'm going to wedge it in there, right? How am I going to draw it? How am I going to represent the cholesterol molecule?
Four.
For what?
Fused ring.
4 fused rings. Good. I'm just going to draw them as circles, not as hexagons. It will take me forever. Is that all right? There's some cholesterol. All right, wedged in there. Oh, and what was the other thing? Glycolipids and glycoproteins. Good. All right, we're getting there. Good. Glycolipids and glycoproteins. What makes them a glycolipid? What makes them a glycoprotein? What's the glyco part?
It's like cholesterol.
A carbohydrate.
Sorry.
Carbohydrate chain. It's all right. You'll get used to this terminology. Draw it a couple of times, say it a bunch of times, you'll get used to it. It's like learning a different language, isn't it? Yeah? Okay. On one of my study abroads to Costa Rica, I was trying to, what was I? Maybe just count to 10. I don't think I got it the entire time I was there. And every time we were in a vehicle, I had everybody trying to teach me. Useless. Biology, though, I'm okay with that. Foreign language, oh. So I do feel your pain if like foreign language is your thing and this isn't. All right. So let's draw a carbohydrate chain attached to this, and we're going to make it a
Glycoprotein.
good glycoprotein. Good. I'm going to draw little circles if that's all right, rather than glucose hexagons. And if I attach it to the lipid, it's going to be a what? Good. See, there is some rhyme and reason, isn't it? There you go. Good. So they're the sort of major things we're going to talk about. Phospholipids and the phospholipid bilayer. We're going to talk about these different kinds of proteins. We're going to talk about, actually I'm not really going to mention glycolipids. I will talk about glycoproteins and I will talk about the cholesterol. So once you can draw this and label everything and understand it, you are good with membrane structure. Pretty much, I tell you. Okay. There we go. Then we've got our peripheral proteins. And then we've got our integral proteins. What else would that integral protein be? What kind of integral protein? Transmembrane. The part that sticks out are the hydrophilic, and the parts in this here, phobic. Good job. Okay, so is everybody okay with structure, membrane structure then? Yep. So now we're going to talk about second concept is plasma membrane fluidity.
Sorry. What is a transmembrane again? I'm sorry.
Transmembrane is simply a protein which spans the entire membrane. It sticks out a little bit on the intracellular side and it sticks out a little bit on the extracellular side.
So what you're pointing at right there, that's the integral?
Both of these are integral.
That's just the one is a trans.
Yes.
Oh, I get it.
Both are integral because they penetrate into this hydrophobic region. But this one is a transmembrane protein because it spans the whole membrane. This one doesn't span it, so it's not a transmembrane protein. Good? Okay. So if I gave you a diagram just like this and asked you to label it, you could label it. Yep? Okay. I wasn't being too subtle there, was I? Okay. All right, so plasma membrane fluidity. What do I mean when I say fluidity?
Moving in and out. Things that can move in and out.
It's aqueous.
Think about it as how runny something is. When I'm talking about fluidity, I'm talking about how runny something is, right? So I want you to think about this. If I got a vertical surface and just tilted it a little bit of an angle, and I dripped some liquid up the top, I could measure how long it would take it to run to the bottom, right? And some liquids are going to run to the bottom very, very quickly. And some are going to take a long time. Think about honey that's cold. It takes a long time, doesn't it? Yeah. Think about water, might do it quickly. But what about honey if I warmed it up? It goes really quick, doesn't it?
Quicker.
So, sorry?
Well, quicker. Right?
Quicker, it does, yeah. Quick is relative, yeah. Would move quicker. So different liquids have different fluidities, right? Did you know that glass technically is a liquid? It just has very little fluidity. In fact, I believe what they've done, they've gone to very old churches and they've measured the thickness at the top and the thickness at the bottom. And there's a little bit of sag in them, I think. I don't know if that's urban legend, as a result of them flowing slightly. But they flow ever so slowly. All right? So fluidity is a measure of how runny something is. All right? So membranes have fluidity, and the fluidity of the membrane is crucially important to how it functions. It must have the correct fluidity to function correctly. Now, does anybody use coconut oil to cook with? All right, those of you who do, you're going to know exactly what I'm talking about right now, right? In the winter, coconut oil in Arizona in a kitchen cabinet is what? Solid. It's white, isn't it?
Yeah.
In the summer, what is it?
Liquid.
It's liquid. That slight temperature difference causes it to go literally from a solid to a liquid, right? Now think about bacon. That bacon right out the fridge, all the fat is solid. It's like lard, isn't it? Heat it up, and it goes to liquid. So temperature can change the fluidity of substances, can't it? Yep. All right. So think about a challenge that a cell faces. The membrane must have the correct fluidity to function properly, but temperature can change the fluidity. So you, on a cold day, your liver is probably at a nice 37 degrees Celsius. Your fingers, on the other hand, might be down to 10 degrees Celsius. Huge temperature change, and you've got to cope with those temperature change differences, and your membranes have got to be kept at the correct fluidity. All right, so membrane fluidity then. The phospholipids in the bilayer can move very freely within the bilayer. In fact, they're extremely mobile some of the time. They can move extremely quickly. It's possible for them to move 2 micrometers or 2 microns. Now that's the length of a very small bacterium in 1 second. That's how quickly they can move in the phospholipid bilayer. They swap places with each other very, very quickly, those phospholipids. Okay? Now, the proteins and most of the lipids, they can drift laterally. So not all the proteins can move and drift, but many of them can. And the proteins move a lot less quickly than the phospholipids. Now, rarely, but it does happen, these phospholipids can flip-flop. Okay? They are able to flip, which means that one that was facing where the head was on the extracellular side, it can flip and then become the head is on the intracellular side. It rarely happens, but there's an awful lot of lateral movement, drifting within the layer. All right, so here's an experiment that was done. It's kind of an interesting experiment to I guess, it gathered data on protein movement within the membrane. So what they did is they took a mouse cell, right? Now it's kind of difficult to get individual animal cells. You've got to kind of chop up the tissue and then treat it in a certain way to sort of break the extracellular matrix connections between cells. But you're able to get individual cells in suspension. Is everyone okay with that? And they did it to mouse cells and they did it to human cells. And then they were able to label or tag all of the proteins in the mouse cell membrane. And they were able to tag all of the proteins in the human cell membrane. So they look different. And then what they did is they hybridized the cells. I'm not quite sure how they do it. I think, honestly, you could probably just shake them or collide them quickly and they would fuse. All right? But anyway, they were able to hybridize the cells. Now, I don't want you to think suddenly we've got this mouse human running around, right? It's just a mouse cell hybridized with a human cell. And what they wanted to see, well, how quickly would those proteins move and mix? Or would they at all? And what they found, in just 1 hour, the proteins were sort of evenly mixed. The mouse and the human proteins completely mixed. So that said that the proteins are also quite mobile within the membrane. Okay? They can move around freely, many of them. Some of them can't, but they can move quite quickly within an hour, completely evenly mixed. And then they did it, let it grow, and they got the big mouse human, which is now in the sewers somewhere in New York, I think, eating the alligators. So. All right, so membranes must be fluid and have the correct fluidity to work properly. If they're too runny, they don't work right. If they're too solid, they don't work right. Because remember, things need to be able to move within the membrane. Now, they're usually about as fluid as salad oil. So think about olive oil or a sort of a thicker oil than canola oil. Yeah, that's about how fluid the membranes are. Maybe even butter on a warm day. Yeah, do you get an idea of how fluid they are? But if you cool them down, they can become a lot more solid. If you warm them up, they'll become a lot more runny. So, as the temperatures cool, they become less fluid. And as the temperatures really cool, just like coconut oil, they can become solid. And they don't work properly. So as temperatures warm, membranes become what?
More liquid.
More fluid. Yep. Okay. Now, if you have a constant temperature, that's not a problem. But most organisms don't live in a constant temperature. So now, what do you think we have to do? What do you think organisms need to do?
Constantly change the regulated body temperature.
So some organisms do. We do, at least our core temperature, but we can't regulate the temperature in our fingers, for example. Like if you're a mule and it's a very cold day, your feet are quite cold. If you're a penguin on ice, your feet are quite cold. So what do you think organisms do?
Change their environment.
So what about a plant? Plant grown on hot rock in Arizona. Let me tell you, in the daytime, 150 degrees maybe, Fahrenheit. At night time, it might cool off considerably. So what must organisms be able to do? I heard something, I didn't know what I heard though, so it all come at me at once. They must be able to regulate or somehow maintain the fluidity under varying temperatures, right? They must be able to regulate that, and they do. So we're going to look at how they do it. All right, so the temperature at which the membrane solidifies depends on the types of lipids. All right, depends on the types of phospholipid and cholesterol. The ratio of saturated to unsaturated fatty acids has a big impact. So at any particular temperature, membranes rich in unsaturated fatty acids. So if you've got lots of unsaturated phospholipid fatty acid tails, they're going to be what compared to those rich in saturated fatty acids? Do you understand what I'm asking? At any particular temperature, a membrane that's rich in unsaturated fatty acids is going to be what compared to a membrane rich in saturated fatty acids?
Are you talking like what molecule?
what's the fluidity going to be?
Oh,
Saturate's gonna be more
less.
fluid.
Which is going to be more fluid?
Saturated is going to be solid, more like
The unsaturated is gonna
condensed. And then unsaturated is going to be more fluid.
We'll do a raise of hands. If it's high in unsaturated fatty acids, who thinks it will be more fluid than one that has lots of saturated fatty acids? Raise your hand if it's going to be more fluid. All right. Those of you that think that if you're rich in unsaturated fatty acids, you will be less fluid than one that has lots of saturated, raise your hand. Those that didn't raise your hand, raise your hand. I saw who you were. If it's a 50-50 bet, always raise your hand to one of them, right? Okay. So we're going to go back to the drawing board a little bit. Do you remember when we talked about saturated and unsaturated fats? Yeah. So we've got triglycerides. Yeah. Triglycerides with saturated fatty acids, they kind of looked like that. The chains were straight and they could pack in very close, couldn't they? And they're the sort of things that are solid at room temperature because they can pack in so close. Good? So they tend to form the solid fats. Oils, on the other hand, have unsaturated fatty acid tails. All right? There's a double bond there, which makes it kink. Now, when there's a kink, they can't pack very closely, can they? Which means they can easily flow past each other. So lots of unsaturated fatty acids means you're going to be more fluid relative to one with lots of saturated fatty acids. Is everyone okay with that? So we don't have triglycerides in a membrane, we've just got phospholipids. But the phospholipid tails can be saturated or unsaturated. Good? So membranes rich in unsaturated fatty acids will be what? More fluid. Good. At any particular temperature. Okay? Is everybody okay with that? Okay. More fluid. Okay. So here's a diagram. On the left, you've got lots of unsaturated fatty acids on these phospholipids. Because they're unsaturated, the tails are all kinked, and they can't pack closely together. So there's not a lot of surface for van der Waals forces to hold them together. They don't pack very closely, so they easily slip over each other. In contrast, if you have lots of saturated tails, they're going to pack really tightly like this, and there's not going to be a lot of movement. So at any particular temperature, this one will be less fluid or more viscous compared to this one, which will be more fluid or less viscous. Good? So now can you come up with an idea about how an organism might regulate membrane fluidity?
Adding more saturated fatty acids.
Yeah, good, good. Sometimes it's going to increase the proportion of unsaturated fatty acids, and sometimes it will decrease them. Just depends on what it needs. Good? All right, we're going to look more at that in a moment. All right, so that's one way that they can regulate fluidity. Change the ratio of the saturated and unsaturated fatty acids in the phospholipids. Good? Here's another way, another little trick up the sleeve, and that's cholesterol. We know that cholesterol molecules are wedged between the phospholipids, and they're going to be found in this hydrophobic region because they are themselves hydrophobic, they're lipids. And cholesterol has an interesting effect on membrane fluidity. It kind of buffers fluidity. Now we've encountered this word buffer before, haven't we? And what do buffers do? With pH.
It
Regulate pH, keeps it fixed, right? Cholesterol tends to buffer membrane fluidity. It kind of keeps it fixed, within limits at least. All right? So, with cholesterol wedged between there, with higher temperatures, cholesterol reduces the fluidity. Now, normally when you increase the temperature, the oil or the lipid would get more runny, wouldn't it? More fluid. Yeah? But when you add cholesterol there, it kind of resists getting fluid as the temperature increases. And that's because the cholesterol kind of stops the phospholipids from moving past each other easily. Yeah? Is everybody okay with that, mechanistically how it works? And on the other end of the spectrum, at lower temperatures, cholesterol stops the membrane from getting too solid. It keeps it more fluid because it prevents the phospholipids from packing together too closely. So it lets them slide a little bit. So does everybody...
Same idea as what was the one before. It takes the balance of the saturated fats and whatnot in the phospholipid tails. Is the same idea.
Well, what it does is it's another mechanism to regulate fluidity. We can increase this, and that makes it more fluid, or we can increase these, it makes them less fluid, right? But that's kind of a permanent way. Do you know what I mean? Not a permanent way, because we can change this ratio. But what cholesterol does is it buffers it. When it gets warm, it stops it getting too runny. When it gets cold, it stops it getting too solid.
How does it do that?
At high temperatures, it stops them from moving past each other too easily.
So there's like more cholesterol? Or they...
Well, you can change the amount of cholesterol in the membrane. You can dump more in or pull more out.
So they don't change them from unsaturated to saturated?
No, cholesterol won't change this ratio. Right? Increasing the number of unsaturated fatty acids has one specific impact, makes it more fluid. This one makes it more solid. Cholesterol makes it less fluid at high temperatures and less solid at room temperatures. Does that make sense? Yep.
So how does it switch and regulate itself between unsaturated and saturated? How does that...
Where is new cell membrane made? Not the emergency room, but the
Endoplasmic reticulum.
the ER, right. So it has to do with what kind of new membrane is made on the ER. Yep. The cell can simply make membrane with more of these if it needs to make it more fluid, or more of these if it needs to make it more solid. Membrane is made on vesicles, it goes and fuses with the cell membrane, and that's how you can change the ratio.
Does it go down to the Golgi too? Does it change?
So I imagine it would, but I don't know. I mean, there's a lot going on. It wouldn't just make a little membrane vesicle to change this. It's going to do a whole bunch of other things to it. So I'm sure it would go via the Golgi as well. It's a good question. All right, so I just want to round this out with this diagram. It's a little better than the diagram I can draw. All right, and you can see the cholesterol molecules wedged in there. And again, this explains the impact of cholesterol on fluidity. Good? All right, awesome. Good.